I've talked throughout the presentation about risks or about determining that this specification might be really critical, and so we're going to want to really look at it closely and make sure that we are hitting it, or it could have a very critical failure mode attached to it if it doesn't work. So we want to make sure that we have a high level of confidence that we're going to pass. And risk management is a whole suite of practices that will vary depending on where you are in the process. In the earlier stages of process, you're thinking about creating clinical use charts and understanding, okay, along the way, where are the areas that I'm imagining that this product could be used that would present the most risk to the patient, that might present the most risk to the person who has to use the product at the points, and that you're thinking about that to help you influence your design choices. But then eventually you use that information to also connect to the design itself. And as you do your design work, you're going to start, understanding more about, well, this design in particular, we see a lot of failures of this tip. We either are going to manage that by having a very tight specification, or we're going to do a lot of inspections, or we're going to, you're probably going to address that to some extent, but you're going to use the information that you're collecting around, well, how frequently do I see certain things happen? What's the potential impact if that happened in the field to a user, to a patient? Do we see a significant clinical issue that could occur if we're delivering, if we have a catheter that needs to be delivered and it's got some kind of a hydrophilic coating on it, and now the product is stuck in the package because humidity got in there? What are the risks that could occur? So you're going to be thinking about those things and using tools like failure modes effects analysis to really analyze and help you determine where you're going to focus the number of tests that you might do for each specification. Some of those things will end up getting a lot of scrutiny because they have either they're occurring frequently or they have a high clinical impact of creating an unsafe situation if they go wrong. So you prioritize that in your testing, whereas other things, perhaps the color of something being exactly white, maybe that's not as critical. So it doesn't necessarily need to be evaluated with the same number of samples. You're not necessarily setting the same confidence goals around achieving an end result that you would if you were focused on something more critical.